In this video, we are going to be concluding the topics in Chapter 16, which is going to be mainly talking about polysaccharides. We are going to be studying their structures and also their functions. So, understand that when it comes to polysaccharides, they are going to be resulting from linking many monosaccharides. All the polysaccharides that we are going to be talking in this section are going to have glucose as the most common monomer, meaning all of these polysaccharides are going to be glucose linkages of one another. So here we have in the structure 3 different polysaccharides, which include starch, cellulose, and glycogen, and we're going to study them in detail. So when it comes to starch, understand that starch is composed of 2 separate polysaccharides. We have amylose, and amylose is actually a straight chain polysaccharide of glucose units, while amylopectin is going to be a branch chain of glucose. As you can see, here we have the structure of amylose, and here we have the structure of amylopectin. So when it comes to this, the only reason why we have glycogen here, which is the next one that we're going to be looking at, is because glycogen is similar in structure to amylopectin. Okay? But understand that glycogen is not present in amylopectin. That is just symbolizing the structure. Now, when it comes to the linkages that are unifying those glucose molecules, in amylose, as you can see, they're going to have an alpha-1,4 glycosidic bond between the glucose molecules. When it comes to amylopectin, you will also have the alpha-1,4 glycosidic bond, but specifically the branches are going to be alpha-1,6 glycosidic bonds. So, glycogen was something that I introduced in the previous slide, but understand that specifically glycogen is a polymer of glucose that is stored in liver and muscle of animals. What does this mean? This is how we store glucose. As I mentioned previously, there are 2 biomolecules that we store for energy. And that is, as we saw previously, lipids, and we store them as triglycerides. And we also store sugar in the form of glycogen. Now, as you can see, if we compare the structures of amylopectin and glycogen, they're similar to one another because we are having polymers of glucose that are branched. It's just that glycogen is going to be highly branched. Now, understand that when it comes to glycogen, it is hydrolyzed in our cells to maintain blood glucose levels and to provide energy in between meals. I just mentioned that it is more branched than amylopectin when we compare the structures, and understand that the glucose units in glycogen are joined by alpha-1,4 glycosidic bonds, okay? And that's in the main chains, and the branches are going to be alpha-1,6 glycosidic bonds. So in terms of how the glucose molecules are going to be linked to one another is very similar to what we observed in amylopectin. When it comes to the last polysaccharide, which is cellulose, understand that cellulose specifically is what gives the rigid structures to the cell walls for specifically things like wood and fiber. Now, understand that this polysaccharides does also have glucose units, okay, but they are not branched. More importantly, understand that when it comes to what are the linkages that are present in cellulose is going to be beta 1,4 glycosidic bonds. In our bodies, we do not have an enzyme that can cut glucose molecules that are unified by a beta 1,4 glycosidic bond. Okay, so understand that when it comes to cellulose, this is not something that we can hydrolyze. Okay, so because this molecule is resistant to hydrolysis, understand that we cannot process it and obtain sugar molecules from it. So that's why we cannot digest it. One of the important features that separates cellulose from other of these molecules is that it cannot form hydrogen bonds with water. It can hydrogen bond with itself, but it doesn't do it with water. So that's why cellulose is actually insoluble in water. And as you can see on the right side of the slide, you can see how specifically these glucose molecules that make up cellulose is going to be making a network of hydrogen bonds to themselves, but they are not going to be interacting with water through hydrogen bonding. So again, cellulose is insoluble. So I know that there's some people out there that say, oh, when you eat things that are fibrous, so for example celery, that is going to contribute negative calories because your body is going to spend more time processing it. That is a myth. It is not true because our bodies are not processing that fiber. Remember, we do not have the enzymes to process those fibrous, that cellulose, I should say. So when you eat celery, all you're obtaining inside the body is just the fiber, which make things being pushed out of the body much better, and you're getting hydrated. So the whole idea of negative calories through celery, it is not correct.